Good day, learners.
Good day, ma'am.
So today we are
Good day,
going
ma'am.
to cover a chapter of DNA replication. But firstly, before I go into details step by step of DNA replication, I'm going to start to explain to you what the DNA is. The DNA is a molecule that consists of two strands are coiled around each other in a double helix shape, and each strand of DNA is made up of a sequence of bases called adenine, thymine, cytosine, and guanine. And remember, adenine is always paired with thymine and guanine is always paired with cytosine. The two strands of DNA molecule runs in an opposite direction, which determines how each strand is going to be replicated. Now let's have a look on a DNA. Let's have a look on a DNA replication by, I'm going to explain to you step by step the DNA replication process together with the enzyme that involved in this process. And DNA replication is the process by which DNA makes a copy of itself during cell division, and this process of DNA replication occurs in a nucleus before the cell divide, so that each new cell produced has a complete set of chromosomes. So learners, how does the DNA replication occurs? The first step of DNA replication The first step of DNA replication is catalyzed by the enzyme called a DNA helicase, which unwinds the DNA double helix, thus separating the strand into two individual strands and creating a replication fork. What is a replication fork? Replication fork is a Y-shaped structure which Y-shaped region on a DNA replicating molecule where a new strand is growing, and with each strand separated, we can now start the process of DNA replication. But before we start the process of DNA replication can start, the enzyme that copy the strands needs to have a place to start, as it is only at nucleotide to the three end of existing strand. So the enzyme called primase bind at a specific location at the DNA and kickstart the replication synthesis, and the primer is run about 5 to 10 nucleotide long. Then another enzyme called DNA replication 3, which is the main replicating enzyme, binds to the primer and begin the process of DNA replication by generating the whole new complementary strand by adding the DNA bases to the chain that was initiated by a primer. Then the DNA polymerase 3 catalyzes the formation of phosphodiester bond that each new nucleotide as is added to the complementary strand. And this process is going to be different in both leading strand and leading strand. And on the leading strand, the DNA replication moves along sorry guys, on the leading strand, the DNA replication moves along with the replication fork, continuously synthesizing the complementary strand requiring only one primer, whereas on the larging strand, polymerase has to go one chunk at a time as the new template is made available, and this chunk is called Okazaki fragments, and they each will require its own primer in order for polymerase to bind to copy a new fragment. After a new fragment is synthesized, the enzyme called DNA polymerase 1 come to the place and will go through the DNA strand and replace the RNA nucleotide from the primer with a DNA nucleotide to make sure is DNA all the way through. Lastly, the enzyme that catalyzes the process of DNA replication is DNA ligase, which joins the Okazaki fragments together to produce a unified strand. So the resulting DNA molecule is consist of newly formed strand and the old original strand of DNA. So I'm going to look on a summary of DNA replication. I'm going to explain it for you, a short summary of DNA replication. A helicase, which is the enzyme that's like the process of DNA replication, start the process by unwinding the DNA double helix and separates the strand by breaking the hydrogen bond between the bases of DNA, thus generating two single strands of DNA, which will act as a template for the synthesis of counterpart strand. And then the second enzyme that catalyzes the replication process is primates, which synthesize small primer, which will act as a starting point before the DNA polymerase 3, which is a replicative enzyme, start the process of synthesizing a complementary strand. And the third enzyme that catalyzes the process is DNA polymerase 3, which on the larging strand, we need only one primer and everything goes continuous. Whereas on the larging strand, we need a primer for each Okazaki fragments formation. So the lastly, DNA ligase joins everything together. So I'm done for today. Do you have any questions where you don't understand so that I can repeat? Lenas, are you still together?
Ma'am, I have a question. Do you mind going back to the differences between leading and lagging strand?
Okay, I'm going to let me explain to you the difference between leading strand. A leading strand is the strand which is synthesized in the 5 to 3 direction, while the lagging strand is the strand which is synthesized in a 3 to 5 direction. Therefore, the leading strand is synthesized continuously, while a lagging strand is synthesized in a fragment called Okazaki fragments. So are you all okay? Learners?
I'm okay, ma'am. Thank you for that clarification.
Okay. So I'm going to leave you with a small piece of homework to go and do it at home, and we are going to discuss it tomorrow in our first period. Here is the question. We'll go and look for it. And tomorrow we are going to start by we are going to start with this question.
Hear me?
Hello.
Can you hear me?
Okay,
Yes,
ma'am.
I can hear you. You have something to ask? Learners? So if you are all fine, I'm going to end my session for today here, and then we'll meet tomorrow. Here is a link. Here is a link. If you don't understand, just go and have a look on this video so that we can have a deep understanding about the process of DNA replication. And tomorrow we are going to start by looking on that homework I gave you. Bye-bye.
Bye, ma'am.
Bye. Have a nice day.
You too, ma'am. Bye.